Welcome everybody to another episode of Dr. Matt and Dr. Mike's medical podcast.
Long form
podcast, unlike those short little 5-minute ones we've been doing, Matt. Hopefully people are still listening to the long form ones. The short form, heaps of people listening. They're loving it. I'm getting great feedback, which is really nice. Today we're talking about a topic which is really complicated, really quite amazing, and personally, I think it's going to be revolutionary when it comes to gene editing and things like cancer therapies.
Oh, so it's not about snacks.
Not about snacks.
Oh.
So
just the name. That's what I thought it was.
Why? What's the name?
CRISPR.
Yeah.
Okay. No.
Okay. Well, we might start the podcast again. So today we are talking about CRISPR, like Matt said. CRISPR is CRISPR. And it often is termed the CRISPR-Cas9 system.
That's what I refer to it as.
Is it? You and your extensive genetic background?
Correct.
Okay.
Correct.
I actually have a PhD in genetics, which didn't necessarily help me with this podcast because it's still quite complicated, and I got my PhD before this really became a technology.
When did you get your PhD? 2015?
15, yes. And while this technology has, well, let's just get into
2002.
it. When they started using this technology?
That was when the name was first coined.
Yeah. Let's jump back a little bit. Let's jump back and talk a little bit about DNA. Let's talk a little bit about...
That's a long way back.
Gene editing. Oh, we don't have to go all the way back. But I think we should talk a little bit about what DNA is, why we have DNA, how DNA is transcribed and translated, and why this is relevant to the discussion of CRISPR-Cas9. So I think we should first begin by saying that the general dogma when it comes to genetics is that we all, in all the cells of our body, except our red blood cells, so all 30 trillion cells of our body, except the red blood cells, which make up a significant number of those.
60% of the body.
Yep.
Yeah.
We have DNA.
Okay.
And the DNA sits within the nucleus of our cells. I think every listener knows this. And we have about 2 meters of DNA in each of these cells. And it's wrapped up tightly in what we call chromosomes.
And we have 23 pairs of those
23
per cell.
pairs per cell. Yep. Yes, that's right. So 22 somatic chromosomes and 1 pair of sex chromosomes or sex-determining chromosomes. But the point we're trying to get across here is we've got a lot of DNA. And the DNA that we have is double-stranded. Mm-hmm. And DNA is made up of nucleotides, which are chemicals. Okay. And
that's the alphabet of them.
It's the alphabet of our DNA. And these include A's, C's, G's, and T's. In what looks like, if you just looked at a cut of DNA, just a random arrangement or assortment of these A's, G's, T's, and C's. Because there's 2 strands, we need to know that on one strand, which we can call the template strand, and another strand which we call the complementary strand, that the base pairs match. Now, they don't match in regards to if there's an A on one strand, there's going to be an A on another. It matches in the sense that A's will always match with a T and G's will always match with a C. So if I've got a template strand of DNA that says A, G, T, C, then the complementary strand will say T, C, G, A.
And so why do we give these terms to a template versus a complementary?
Great question. It's because that when we want to... Well, the purpose of the DNA is that it...
holds information.
Yes. So people say the blueprint of the body, which in part is quite correct, because the DNA needs to be transcribed.
So read and changed.
Yes. So for example, if I asked you to just transcribe this paragraph I wrote, it's asking you to just look at it and copy it out.
Right.
So the body needs to do that with
copy.
DNA. Direct copy of DNA. And it needs to do that by copying one of those strands, often the template strand. And because
is that always the case?
No, it's not always the case, but the general dogma that we talk about in genetics is that the template strand is used as the template to make a copy.
Right.
And the copy that they make ends up being obviously what looks like identical to the complementary strand.
Okay.
So DNA being double-stranded, bound together, it opens up. We read the template strand. It might say AATC, and the body goes, I'm going to write that down as TT. What did I say? AAGC?
Something like that.
TTCG. right? Because that's the complementary. And now I've just transcribed the template strand. What we've just made is something called RNA, a single strand of complementary DNA or RNA, which is complementary to the DNA. There's obviously a couple of differences. For example, it doesn't have T's, it has U's for uracil. But the point of this RNA strand is that it gets read by proteins called ribosomes, turned into amino acids, and they fold into functional proteins.
So then if you want to make a protein from your DNA,
Yes.
is that always coming off the template strand?
Not always.
Okay.
No. But again, the general dogma that we read in textbooks is that it is.
It is.
But no, that's not necessarily the case. So,
is there any reason for an exception?
well, it's just the biology of it. So, for example, sometimes if you, so when I was doing my PhD, for example, and you would look at DNA strands that we know would encode for genes, the first thing you need to know is that DNA has what they call a bunch of dark DNA. It used to be called junk DNA, where we didn't know what it was. It was sort of just strands of these A's, T's, G's, and C's, but it didn't correspond to a gene.
When you say gene, do you mean like a functional outcome?
Yes. So
it just seemed to be like it's there, but it doesn't serve any purpose.
This dark DNA. So just remember that a gene is the minimum amount of RNA we need to...
RNA or DNA?
RNA.
Oh, okay.
But it can be DNA.
Okay.
The problem... So let's just go back another step. When you've got a big long strand of DNA, you could say, okay, from here to here, from this DNA, it's going to ultimately encode into a gene.
Yeah.
But there's going to be stretches within that DNA that need to be cut out, called exons.
Okay.
Right?
Yep.
And then what's left are the introns, and they sort of snap together.
And they'll be expressed.
And they will be read into the RNA.
Okay.
And that RNA will then be expressed into a protein. So what we have here is a gene that can be read into a protein, and protein has function. You're asking about the way it gets read. So you can read the template, 5' to 3'. So that's from one end of the strand to the other. Or you can read it 3' to 5', or you can read the complementary strand 5' to 3', or 3' to 5'.
And then puzzle it together.
Yeah.
Okay.
Yeah. And, sometimes, there's a lot of complexity with it, because sometimes you can have one stretch of DNA and it can encode the same gene, but a variation of that same gene, because it decides, I'm going to pop that exon out, but I'm actually going to leave this exon in if I want to transcribe it into that gene, right? So there's a lot of complexity with DNA, but I think we need to distill it down to the basics, being human DNA is double-stranded.
Yep.
It's made up of A's, G's, T's, and C's. When we want to make a protein, and proteins do everything in the body, we need to turn that DNA, transcribe it into RNA, which is single-stranded, and then read it and translate it into a protein. And again, that protein does everything. Now, the thing is that then states that the protein that we make is determined by the sequence of nucleotides in the original DNA strand. So if you were to change those nucleotides, you may change that protein. Now, it may turn the protein off and it no longer works because the change in the DNA meant that a functional region of the protein no longer exists. Or maybe it adds a function to this protein and now it is overactive. So there's possible different outcomes by changing the DNA. And we need to be mindful of that when having the conversation about CRISPR.
Okay. So this then is part of the reason why we want to employ this gene editing technology, where we could play around with the way that the genes are not manipulated in a way, but we could potentially edit them. So if you were to write a document, again, you're using the English alphabet, which has how many letters? 26? 26, there we go.
So I probably should know that.
And so if you, let's say you made an error with one type of word and it was a reoccurring error, what you could potentially do in that document is, let's just say, do a Control F function and find that word, and you could slightly either extract the whole word out and exchange it for something else.
Yep.
Or you could do a letter manipulation.
Yeah, just change it so it fixes the word.
Right. And so CRISPR in a way is a similar approach where we can now use, but in this case, the alphabet of the DNA is only 4-lettered.
Yeah.
And we can look for genes, which is, let's say, equivalent to the word. And we can slightly modify that to make it either turned off or becoming more functional.
So you could turn it off, you could turn it on, or you could change it entirely. And so what that then tells us is that CRISPR is gene editing.
So when we look at genetic disorders, sometimes we might hear these terms of a dominant gene mutation or disease, or we might have recessive. Can you quickly just tell us the difference between what a dominant...
Yep. So the first thing is that we should be aware that not all diseases and disorders are genetic in their origins or basis.
Right.
And that's important because it means that gene editing technology like CRISPR is not going to be a curative for all diseases and disorders, only those that that are genetic in their origin. Now, to go back to your question about the way certain genes are inherited, we need to understand that while all the DNA in our body is double-stranded, we have 2 copies of this double-stranded DNA. One we got from mum, one we got from dad. So effectively, every gene that you have in your body, for example, and I'm going to oversimplify this to the point that it's incorrect, that the gene that you have in your DNA that states that you have blue eyes, right? And now it's obviously...
There's a multitude of them.
That's right.
But let's just say there's
Let's
one.
say it's one. You have 2 copies of that because you would have got one from mum, one from dad. Now, the thing is that most of the time, these genes are actually identical, the ones we get from mum and dad. But every now and then, there's some slight differences. A single base pair or nucleotide is different, or maybe multiple base pairs or nucleotides are different. And that may or may not change the way that, like we said earlier, gene is translated into a protein and whether that protein has function. Now, there's 2 types of ways that we can inherit these types of genes. We can have a dominant inheritance pattern or a recessive inheritance pattern. Because we have 2 copies, the way I like to think about it is a recessive inheritance pattern. You need 2 copies of that gene to express the phenotype.
Right.
So now I've just introduced the word phenotype, which means the way it looks.
Seen.
When I look at you right now, I'm looking at the phenotype, the way it's expressed.
So the blue eyes, you can see the blue eyes.
Yes. So a recessively inherited gene means you need to have 2 copies of it in order for it to express the phenotype. A dominantly inherited gene means you only need 1 copy of this gene in its form to express the phenotype. And the other one, in a way, is quite redundant.
Okay.
So that's important because then when you start talking about not just the way traits are inherited, but the way diseases and disorders are inherited, some are recessively inherited, some are dominantly inherited. And that's important because when it comes to recessive inheritance, you've got a backup gene, right? So that is also important because a lot of the times if we have a mutation in one gene...
And it becomes dysfunctional, you have a backup.
That's right. That's right. Unless it's dominantly inherited.
Yeah.
Now, if you have a mutation in one gene...
Can you give an example of a disorder?
So think about it like this. Usually, dominantly inherited disorders is what we call a toxic gain of function. So the mutation that's present in that gene results in a disease or disorder that
overexpresses,
has changed the protein in a way that it's now a toxic gain of function. The protein works, but in a way that's detrimental to the organism.
Okay.
And it only needs to be in one of those because it's so bad that it doesn't matter that the other one's good, it's overriding the good one.
Okay.
So, it's sort of like you and I. Let's just say I do the right thing, you decide to do the wrong thing all the time, you start to smash things up. Doesn't matter if I'm still sitting here doing the right thing, you're still smashing the room up, right? So all people see is you...
A broken room.
Breaking everything up, right? So that's a toxic gain of function. But with recessive inheritance, the mutations that are present usually inactivate the gene and result in a loss of function. And this is why the redundant or spare gene is important, because again, if you and I are these genes and there's a mutation in me and I stop working, I just sit here, you can still keep doing the podcast. You can keep talking. And so there's redundancy in the system. So,
and is that like with an example of a recessive disorder, then sometimes it doesn't become present, so it doesn't become phenotypic until, let's say, later in life. Is that kind of because its kind of function wears out?
Yeah, so there's something called haploinsufficiency. And so sometimes there are, so Parkinson's disease, for example, there's a couple of genes that are recessively inherited and with certain mutations in them can result in Parkinson's disease. But often, everything looks fine. There's no problem. The phenotype seems absolutely normal, quote unquote. And then later on in life, they present with this disease or disorder because there was a problem, but the backup gene is able to make up for the issue. But then over time, it's not enough, right?
Okay.
It's almost like it loses steam.
Yeah, yeah.
And then the disease is able to manifest.
Right.
Yeah.
Okay. So before we get, is there anything else you wanted to discuss?
No, I think that we just needed to preface these concepts because the way CRISPR works, and we'll get into the nitty-gritty in a sec, is that for genetic diseases, it could be genetically inherited. So it could be in a germ line, which means sex cells.
So an example of that could be haemophilia.
Yes.
And so that would be, it's an X-linked genetic alteration.
Yep.
And because females have two X's, they generally, if they were to have this faulty gene, they have the backup to overcome it. And so then they may not phenotypically demonstrate it.
Yes.
But because males only have one X chromosome, then that's it for them.
They don't have that redundancy like they do with all the other chromosomes.
And so then they would not only have it in the genotypically, but they would also express it.
Yes, that's
phenotypically.
Right. Yeah. So I think that's really important for us to understand is that CRISPR and other gene editing technologies, but CRISPR is the paramount one and obviously the topic of conversation today. And I think what you often hear the conversation when it comes to CRISPR, focusing on cancer. And the reason is because cancer by definition is genetic. It's based in our genes. Cancer arises because mutations occur in very specific genes, often two major types. Tumor suppressor genes, which turn cells off and just say, hey, stop growing, and oncogenes, which turn cells on and say, hey, keep going. And often the mutations in the tumor suppressor switch it off, so it can't stop the cell from growing, and the mutation in the oncogene is a toxic gain of function, right?
Yep.
And turns it on and keeps it going. Now, there's obviously other genes that sort of work in and around that promote metastasis and tumorigenesis. And obviously, these are going to be mutations in genes that produce proteins that can change the way that a cell metabolizes a nutrient or the way that the blood vessels grow into a tissue. Like, there's obviously other things that happen to promote tumor growth, cell growth, and so forth. But it's all in the genes. And so the purpose of CRISPR is to be able to target these particular genes and change them and fix them so that it limits, stops, halts the growth of these cancers.
Right. And so even before we get to CRISPR, there were some gene editing technologies that were utilized.
Yeah.
But they were more rough and ready, I guess you would say.
Yeah,
so
very much.
I'm not really sure, you may know this, but I'm not really sure in what era humans started to play around with genetic coding and so forth.
Mendel.
Like 50, what was in the 50s when we discovered DNA officially,
yeah, but
right?
Mendel the monk back in the 1800s was playing with the genetic makeup of pea plants.
Well, that brings an interesting point up. So, and I know you've had pushback with your students on this at times where we generally think, oh, look, when we genetically modify things, this is us, playing God, essentially, right? But essentially, we've been doing this since we're humans, right?
Oh, absolutely.
So, I mean, from my understanding, at least domestication, possibly the first domesticated animal was the wolf.
Yep.
At least from my understanding, that's something that's been evolving with us alongside our own kind of adaptation evolution.
Yeah, we've genetically modified
possibly
wolves.
30,000 years running beside us, literally.
Yeah.
But we have, plants, wheat, bananas.
Oh, look, there's probably not a fruit or vegetable that you ingest that isn't genetically modified. And I think the point we need to raise here is that genetic modification, the thought that it starts to bring into people's minds is a scientist in a lab sticking a pipette into a banana, right? That's not what's happening. I mean, you can genetically modify plants by crossbreeding them.
Yeah, so that's going to what you were talking about with the monk.
Yeah.
So the monk in, when did you say it was?
1800s.
Basically,
Gregor Mendel, the monk.
Basically looked at the phenotype of these pea plants by the colour of their flowers, and then got a little paintbrush, and got the sperm of the animal, which is the pollen, and then rubbed it on the ovum of other plants to see how this may...
plant and animal, do you?
Oh, did I say animal?
Yeah.
Sorry. The ovum of the other plant,
yeah,
and to see what effect that would have.
Yeah, he genetically modified them.
Right. So then there was an idea of these outcomes being what would occur.
Yeah.
But then we started, when we had a better understanding of what DNA was, which again, Crick and...
Well...
there's three of them. Correct?
Yes.
Yeah.
People tend to focus on Watson and Crick, but it wasn't
right.
really.
At least there was a third as well.
Yes.
Now, then there were other things that we were doing with DNA randomly, where we would just, get seeds and then X-ray them, and that would just put mutations into the DNA, and then they would see what kind of outcome would come about from doing that, and then see if there was an improvement or something.
Yeah. Let me just add Rosalind Franklin.
Yeah.
Watson, Crick, Rosalind Franklin. Rosalind Franklin had the X-ray of DNA, didn't necessarily recognize what it was, showed it to Watson and Crick, and they went double-stranded helix. Sorry. Just wanted to say, because everyone talks about Watson and Crick when they were only part of the discovery. But continue. I apologize.
So then, as we got more understanding of what this code was doing, we started to try to manipulate it more accurately. And so pre-CRISPR, we would start to do things more rough and ready, where we would chunk bits out and throw it in.
Well, we didn't even know what we were doing. They were all guesses. We didn't know whether the genetic changes we were making were going to result in, let's just say, a fruit that would have a seed so big that there was nothing edible present, or whether we were making a fruit that tasted better or worse, or was toxic or not toxic. And I mean, it was a blind approach to playing with the DNA of the plant. Yet, because people see it as quote unquote natural, because all you were doing were transplanting aspects of one plant to another and crossbreeding them, and that anybody could really do it, it was, oh no, it's natural, it's fine. But in actual fact, what we do in the lab today is far more precise and specific.
Right.
We don't just put our blindfolds on and just take a bit of DNA and throw it in and hope for the best. We look at genes that we know translate into very specific proteins that have an important function, and we can take that gene and we can put it into another plant, no different to what we've been doing for centuries, right? But we're just more specific and direct about it so that we know what the outcome will be. So when people lose, and I think it's happening less and less and less, but when people lose their mind about genetic modification of foods, honestly, everything you're eating is going to be, in one way or another, genetically modified from its origin. I mean, you and I are genetically modified, right? I mean, that's just what biology is.
Yeah.
Our genes are constantly being modified by our environment and our interactions. We're not this static thing. My DNA isn't identical to the very first organism that existed on this earth. However, it did originate from it, but it's very different.
Right.
I mean, we're more related to fungi than fungi are to plants, right? So it means we have a common ancestor.
Right.
But are we the same? No, we're not the same. We share 60% of our DNA with a banana still, right? So
speak for yourself.
Well, yeah, you more so. 60% of your DNA with a gherkin, or maybe a squash.
Anyway, I just want to quickly
want to quickly
highlight the pre-CRISPR gene editing days, which was around the late 80s, 90s,
Yeah.
what they basically did was they isolated the gene that was causing the potential change in the phenotype of the individual. It could be like certain biochemistry issues like ornithine, which
What? The amino acid.
ornithine, ornithine, which would result in the way that biochemistry the person would process ammonia. So that in kind of within the urea cycle in the liver, if they had a deficiency in this gene, they basically couldn't clear ammonia, and that would become toxic.
Yeah.
And so some of these conditions, they tried to insert a whole chunk of a functional gene into an individual cell's ex vivo, which means outside the body.
So they took the patient's cells, put it into a dish, and then did it in the dish so they could reintroduce it back into the body. Yep.
And so some of the downstream effects of this, or off-target effects, which kind of put a halt to this gene editing approach, was either it would stimulate the immune system, and at least in one case, I think the patient was Jessie, 18-year-old female, who died within about four days of reinserting her own cells.
Because of a huge immune response.
Yes, that's right.
And the immune response was because it had a bit of DNA added that it recognized as non-self?
Or it could be partly because we introduce it through viral vectors. So we use viruses.
So explain what a vector is.
A vector is, I guess, something that it allows us to inject the gene the gene or the DNA into the cell itself, the host cell.
Yeah, okay. It's a way of carrying things, generally speaking, but here information or DNA. And so, for example, just to interrupt, people will often call mosquitoes
a vector
for malaria.
Right.
So it's a carrier of malaria to help introduce malaria into another organism. And there's many types of vectors that we can use in biology, and particularly gene engineering and so forth. One of which is viruses, because we know viruses are really great at introducing DNA into organisms. And now they're inactivated viruses, by the way. So they take things like adenovirus.
And that actually was the approach that they used with one of the vaccines. We used a viral vector.
Yeah, well, that's actually very common, isn't it? Yeah. And so you take the virus, you inactivate the virus in the sense that you can still use it to introduce DNA, but it has no virulence. So it means that your body's not going to translate any of the viral genes into something that causes a disease, right? So it's inactivated in that sense, but it still introduces the gene that you want. But because it's viral and exogenous and comes from something else, your body may recognize it as foreign. You may have an immune response, like you said earlier. Yeah. The other type of vector that we often use in biotechnology or within genetics, molecular biology, whatever, are plasmids. So plasmids are actually bacterial DNA, or actually part of bacterial DNA. They're sort of separate to bacterial DNA. They're sort of standalones, but they're circular aspects.
Is this how they share?
Yes.
This is how the bacteria share genes amongst each other?
Yeah, so the way humans share genes are through sex and having offspring, and the way that bacteria share genes are through plasmids. So they can actually what we call translocate their own DNA and say, hey, I've got this gene, you may want this gene, and they literally hand it to the other bacteria that incorporates it into their genome.
And that could be a good example of how bacteria become resistant to antibiotics.
Absolutely.
You might have a slight not alteration, but there's difference within a small population of bacteria in a patient that has resistance to penicillin.
Yeah.
And they may be the only bacteria that survive that dose of penicillin, but they can share that plasmid with their neighbours and say, here is what you need to overcome the penicillin. Yeah. And then all of a sudden you have a big population of bacteria that is now resistant to penicillin.
Absolutely. So we can take these plasmids and we can, because it's a circle, we chop it and open the circle up and we add our own gene into it, close it back up again, and then we can introduce that plasmid into a cell or body or whatever it may be. Generally, we're introducing it into cells in a dish, but you can introduce that into an organism so that it expresses that gene.
Right.
So these, like you said, these are
these
the
were
more traditional.
Yeah. So
ways.
the two big drawbacks with the pre-CRISPR approaches were the immune toxicity,
Yep.
but also the oncogenesis. So because we're just chucking big chunks into the genome, what could result potentially is that we create a oncogene.
Yes.
And it then makes, and in these cases, another example of these early 90s were patients would develop leukemia.
Yeah. Yeah.
And so this kind of, I guess, put a halt on things until they looked at alternative approaches. Instead of doing the big chunk in DNA, they looked at maybe slight manipulations.
Yeah.
And this led to how we first understood what CRISPR was, right?
Yeah. And I think, like Matt said earlier, like you said earlier, is the traditional forms of gene editing is like taking a saw to the genome and just chopping it, right? It's sort of rough, ready. You might damage it more so than you wanted to. But then when you compare it to CRISPR, what we have now, it's like taking a scalpel to our DNA. It's very specific and fine and precise. And so is it now time for us to really, 30 minutes into the podcast,
can you
talk about CRISPR?
Can you introduce where it was discovered?
Sure. So back in 1987, the year I was born, a team in Osaka, Japan, what they saw when they were looking at E. coli was that there were these... The bacteria, you're right. They saw that there were these stretches of DNA or segments of DNA in the bacteria, around about 30 to 40 base pairs long, they repeated and they were clustered in very... Sorry, microphone just dropped out. They were clustered and into very specific areas. They repeated and they were palindromic.
What does that mean?
They were read the same way forwards as they were backwards, like kayak or race car, right? So they're clustered, they're regular, they're repeating, and they're palindromic, and they were interspersed or interspaced with other segments of DNA which they had no idea what it was. And so they didn't know what this thing was. They published this in 1987. They didn't give a name for it, really. It definitely wasn't called CRISPR then in 1987. And then it took about a decade after that, even more, I think about 15 odd years, before researchers looked at it and went, hey, these aspects of DNA, these are clustered, repeating, palindromic segments of DNA. That's actually not the interesting part. They were focusing on that, came to nothing, and then realized, let's have a look at the spaces in between these repeating bits of DNA. And they realized that there's this program that I use throughout my PhD called BLAST, where you can take a DNA segment and you paste it in to the computer, to this database. You press the button BLAST, and it compares that DNA section with every organism on the planet that has been genetically screened.
Right.
And it comes up and shows you other organisms that it's similar to. So what they found was that these spaces in between the repeating spaces, they matched to fragments of viral DNA. And they're like, wow, okay, so we've got bacteria containing 20 to 30 base pairs of viral DNA, and either side it's surrounded by these repeating, clustered, palindromic sections of DNA. Interesting. They then started to screen this in more bacteria and more other types of organisms like archaea, and they found that these sections of DNA were present in up to 50% of bacteria and 90% of archaea.
Okay.
So they're very common. And what they called these were CRISPR. And CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. And it makes total sense. The clustered areas, so it's not spread throughout the entire genome of the bacteria or archaea, it's in very discrete, specific areas. They're regularly interspaced, so you've got the palindromic repeats there in the name, interspaced by bits of DNA that come from viruses. And you call that CRISPR. Now, in actual fact, some bacteria and archaea can have hundreds of these interspaced things that we call spacers. So the viral DNA in the genome, we call it a spacer.
Right.
You can have hundreds of these spacers.
Would the bacteria use those for any reason?
Yes. So if let's just say it's got 100 spacers, it means that it's effectively stolen fragments from 100 different viruses, right?
Right.
And it's incorporated those fragments into its own genome.
Okay, so...
And its purpose, which you asked, is to remember the virus that had previously attacked it.
Okay, so one quick step back. For a virus to become successful in its reproduction, because we generally would consider viruses to be not alive.
Yeah.
Because they don't conduct biochemistry or can reproduce on their own.
Yep.
All viruses are generally are packages of genetic information, whether they are RNA or DNA, but they would infect a cell, and that could be human cells, animal cells, plant cells, but also bacteria.
Yep.
And they can insert their genetic information.
And that virus is generally called a bacteriophage.
Okay.
If it infects a bacteria.
All right. And then it can tell the bacteria to reproduce its machinery for it to then have its offspring. Is that correct?
I think that's great. Viruses are packages of DNA which it can't. Now remember, we spoke about earlier, DNA needs to go to RNA, which then needs to be translated into functional proteins.
Yep.
And basically, if you look at viral DNA, it's the blueprint to make more viruses.
Yep.
And viruses are functional proteins. Right?
Okay, right.
So, but it can't make itself. So like you said, it hijacks the machinery of whatever organism and then says, in this case, the bacteria, hey, transcribe my DNA into RNA, then translate it into a protein, and then it repackages itself into a virus and then legs it and goes to other bacteria or other organisms.
And would you assume that, I know in humans it generally would result in the cell either dying on its own or being killed off by the immune system.
Yeah.
But would a bacteria or an archaea die from
a viral infection?
Yeah.
Yeah, absolutely.
Okay.
Yeah.
So basically what we're seeing here is a kind of like a historical map. If we look at the genome of these bacterial archaea on what viruses their forefathers have been.
It is a memory of all the viruses that has infected it in the past and remember, a bacteria isn't like us. It doesn't have a lifespan of 70, 80 years, right? It may have a lifespan of minutes to days.
Okay.
Right? And so in actual fact, a bacteria is as successful as its offspring. And so it doesn't replicate through sexual reproduction like we do. It's through binary fission. It splits in half, just makes copies of itself. So effectively, its offspring, like you said, its forefathers and its offspring will have basically identical DNA. And so the bacteria today that we have will have a genetic memory of the viruses that infected it, possibly tens of thousands of generations, millions of generations ago, right?
So these sections of the bacteria archaea's genetic blueprint are chunks of the viral RNA DNA.
Yep.
And what
spaced between them, you have the repeating palindromic segments of DNA.
And so why would a bacteria want to hold on to that?
Wants to hold on to it because of the whole CRISPR system. And what it does is it doesn't just remember it, because that's pointless, right? It's like saying, oh yeah, I remembered my attacker, but then when you see that attacker again and they just attack you and knock you down, you're like, yep, that was him. You know, it's unless you do something about it, you need to be able to recognize and then respond. And that's what this does. It allows for them to recognize and respond, and the response is through this CRISPR-Cas9 system. So just to go back one more step, I said that when a bacteria takes a fragment of the viral DNA and incorporates it into its genome, once it incorporates it into its genome, after that incorporation, it puts one of those repeating palindromic segments. And then, if it gets infected by another virus, it puts it in after that previous palindromic segment, and then it puts another palindromic segment. So then you've got these interspaced, what we call spacers, that's the viral DNA, the spacers, flanked by repeats. And like I said, and that is what we call CRISPR. Now you can have hundreds or tens of them or whatever, and they're called CRISPR arrays. So bacteria and archaea will have CRISPR arrays in their genome. And we didn't realize this until it would have been maybe 15, 20 years ago. And they first discovered it really in like food biotech. So did you know that?
Yeah, I've got 2002. The CRISPR term was first coined. 2002.
There you go. And it was utilized not as a necessarily gene editing technology, to be able to effectively, and we'll talk about how it changes DNA, but it was utilized in cheeses and yogurts as a way to improve the process because you need bacteria to make these products, right? So, but the rate limiting step was bacteriophages were killing off the bacteria as they were making vats of yogurt and cheese.
Okay.
So they use CRISPR technology to just keep introducing new viral fragments to make them immune so that they couldn't die off and you could just keep making more cheeses and yogurts. And every single cheese that you ingest today on this planet has been produced through some sort of CRISPR technology.
Wow.
Just so you know, whether it's on a cheeseburger or whether it's, grated onto
cheese
pasta
platter.
or a cheese platter. But we haven't spoken about how it actually works yet, right? So how it's useful. So a couple of things. First thing is that when a virus or a bacteriophage in this case, so let's talk about a bacteriophage attacking a bacteria. It comes in.
They look like a spaceship, kind of.
Yeah,
like,
they do,
like
don't
the
they?
moon landers.
Yeah, it looks like a moon lander. And right at the bottom of the moon lander, it injects its viral DNA, which often is double-stranded DNA. So it injects it into the bacteria. The bacteria has two proteins called Cas1 and Cas2, which stands for CRISPR-associated protein 1, CRISPR-associated protein 2. These two proteins are enzymes, and they bind together, and they now form molecular scissors. They will go to the viral DNA, and they'll chop it, and they'll chop a fragment out. The question is, how does it know where to chop? It always chops at an area upstream of a very specific sequence of DNA called a PAM, which is a protospacer adjacent motif. So basically, it scans the viral DNA until it finds this very specific sequence and goes, there it is, and then it counts maybe 4 or 5 nucleotides upstream of that, and then chop, and chops a fragment out.
Okay.
And so now what we've got is a fragment of viral DNA around about 20 to 30 base pairs long, and then it incorporates it into the double-stranded circular bacterial genome.
Okay, so is this in conjunction to the virus being replicated in the cell as well? So is this happening simultaneously?
Yes. Similar to when we get infected by a virus, we are both suffering the consequences of the infection from the virus replicating, but at the same time, we're creating a memory of that virus so that next time we get infected, we don't get as sick.
So in a way that this is the bacteria's adaptive immune system.
Perfect. It's exactly what it is. It's the bacteria's adaptive immune system, which we never thought they had, right? We thought everything was sort of like our first line of defense, our innate immune system, where it's just like
non-specific.
Yeah, didn't care what was infecting it. It would just attack it and kill it the same way. Not the case.
Okay.
It was very specific depending on what virus infected it. So now we've got Cas1, Cas2, chops the viral DNA, steals this fragment of DNA, which we now call a spacer, right?
Yep.
When it chops it in the viral DNA, it's called a protospacer. But then once it takes it and incorporates it into the bacterial genome, it's called a spacer.
And how does it know where to put it in its own genome?
Those PAM regions,
okay,
right? So it incorporates it and puts a repeat behind it, one of those clustered palindromic repeats.
Yep.
Now we've got the fragment of viral DNA incorporated into the bacterial DNA, into the double-stranded DNA. So the next step that happens is that we need to transcribe that double-stranded bacterial DNA, the same way we do for ours, right? So we need to turn it into RNA. And again, you take one strand, a template strand, and you copy it, and you create a complementary strand of it. And so what happens is you have these RNA polymerases that come in, and they make a copy of the template. They create a complementary RNA strand of the entire CRISPR array. So it's made, let's just say, a repeat, a spacer, a repeat, a spacer, a repeat, a spacer, a repeat. And so you got this entire CRISPR array, and we call it pre-CRISPR RNA. That's what that's called. Single-stranded.
Yep.
Now, here's the thing, a really cool thing. The bacteria has more RNA polymerases, right? Goes to a totally different area of its genome and transcribes more RNA. And interestingly, this RNA that it transcribes are very small fragments, and this RNA has stretches of it. Let's just say if it's 100 base pairs long, it will have maybe 15 nucleotides that match perfectly with the repeat, right? That CRISPR repeat, the palindromic repeat.
Okay. Yep.
And then it will have another section that doesn't match, but then maybe 15 base pairs down, it has 20 base pairs that match as well. So it sort of loops in and has parts of this long strand of RNA, parts that bind to the repeat and parts that don't. And it just ends up having this loop DNA with parts of it attached and other parts not. This is called tracer RNA, TRACR, similar to CRISPR, CRISPR. So this tracer RNA is now bound to the repeat, not the spacer. It's not bound to the part that has come from the viral DNA, just the repeats. So what you now have is a pre-CRISPR RNA strand, this really long strand with spacer, repeat, spacer, repeat, spacer, repeat, and then attached or bound to the repeats is tracer RNAs. So you'll have one RNA for one tracer, then you'll have another RNA for, one tracer RNA for one repeat, another tracer RNA for another repeat, and another tracer RNA for another repeat. Now, what we have is we need an RNase, a pair of scissors to now chop up this big long pre-CRISPR RNA with the tracer RNA attached to it, and it chops it up so that and this is the thing that's very cool, it will chop up this pre-CRISPR RNA so that it has a full spacer, only part of a repeat, with a tracer bound to that repeat part. And it does that for the entire pre-CRISPR RNA. So it creates all of these spaces with a part of a repeat with a tracer RNA molecule attached. That's now what we call CRISPR RNA. So CRISPR RNA is 2 RNA strands. One RNA strand is a full spacer, so a full bit of DNA that comes from a virus with a little bit of a repeat, and bound to that repeat is the tracer RNA. That's the second piece of RNA. Now, what comes along is a protein called Cas9. It incorporates these 2 RNA strands into its core. Now, picture Cas9 as like a bean, right? Like a kidney. Now, we know that what's the part of the kidney that's got the internal smaller flexion to it, the smallest edge, shortest edge?
You asked me this question?
Yeah, like the internal, I'm trying to think the best way to explain it to the audience. If you've got a bean.
You mean like the hilum?
Yes, like the hilum. So if you take a bean, you're obviously going to have...
the concavity area.
Yes, the concave part. Okay, so picture Cas9 like that bean. At the concave part, the spacer is sticking out of it. So it's not embedded in the Cas9, it's just sticking out. But embedded in the bean, embedded in the Cas9, is that part of the repeat and that tracer molecule. Now your question might be, what the hell is the point of the tracer? It anchors the RNA strands into Cas9.
Okay.
I heard, and I wish I remembered his name, but I heard a lecturer say that it's like having your glasses, where the arms of the glasses which attach to your head, that's the tracer RNA, but then the repeat and the protospacer is the rest of the glasses in the frame.
Okay.
Right? Or the lens and the rest of the frame. Now what happens? Well, what happens is we've got the spacer hanging out, and the spacer will be complementary to a viral DNA. So next time a virus attacks, this Cas9 protein plus the CRISPR RNA plus the tracer RNA together, they're the 3 things you need. This is why it's called the CRISPR-Cas9 complex or system. It can go to that viral DNA that is attacking you. The spacer binds perfectly with the viral DNA.
As it's getting injected in?
Yes, for the next infection, right?
Okay.
The spacer binds perfectly with a complementary aspect of the viral DNA. That's great. And then the Cas9 protein will snip, and cut both strands of the viral
Viral
double-stranded DNA.
which inactivates it.
Yes. Now, the thing is, a lot of people say, well, why does it inactivate it? Won't it just fix itself? All DNA can fix itself. Not the virus, right? It doesn't have its own machinery. So when you chop a virus up, it's not going to fix itself. So it's gone. It's effectively useless. And that's the immune system. That's the adaptive immune system for bacteria. That's how it works. Now, you can probably sit back and go, okay, I can sort of see how we could hijack this for mammalian cells, our cells, to make changes. So I want you to think about this now. Let's just say I were to introduce this CRISPR. No, let's go back a little bit. I said to you that in the Cas9 protein, you've got two RNA molecules, right? You've got the CRISPR RNA, which is made up of the spacer and a part of the repeat, and the tracer RNA. And they're just bound through hydrogen bonds, right? The same way one DNA strand is bound to another. But what you can do is you can actually make them all one RNA molecule by adding a few base pairs to link them up. The blunt ends of them, you could link them up together. So they're just one bit of RNA that's just looped in on itself.
Okay.
You can do this in the lab. When you do this, you create one single guided RNA called an sgRNA. And so that means in the lab, I can actually create my own spacer, repeat, tracer RNA to go in Cas9. And that means if I can make that spacer anything I want, I can direct it to any part of the genome and chop any part of the genome. Double-stranded. Perfect. But this is the important part of it, is that if I were to do this in any one of your cells, Matt, you have the transcriptional machinery to fix the break. So we've got a double-stranded DNA break. There's two main ways that your cells can fix a double-stranded break. One is through what's called non-homologous end joining. And this is simply where you just glue those ends back together. Now, often, if you just glue those broken ends back together, the transcriptional machinery inside just will either add a random nucleotide or take a random nucleotide or maybe just take a couple or add a couple. And many times this inactivates that piece of DNA. So
the gene, like
that portion of DNA, if that sits within a gene, then that gene's inactivated. Right? Because you've either added nucleotides that are no longer compatible with that gene, or you've removed nucleotides, which means, again, that gene's no longer compatible to turn into a protein. It's inactivated. So many times...
Would that be termed a frame shift mutation?
It would be a frame shift if something was added or removed that then altered the way that the amino acids read. Because as the listener is probably aware, we read amino acids 3 nucleotides at a time. If you remove one, then you've shifted the reading up or back. And if you've added one, you've shifted the reading up and back. So everything after the place that you've added or removed, every one of those amino acids will likely be different. That's called a frame shift mutation, which very much so would change that protein, often inactivate it. So non-homologous end joining with CRISPR often inactivates genes.
Yes.
And the term we use for this is called a knockout. So you can, for example, if I go, and we're not using it like this in biology, by the way, this is just as an example. If I go, that gene that makes your eyes blue, I'm going to turn it off. I put this CRISPR-Cas9 in. I know that the RNA molecule, the single guided RNA I've made, that's embedded in that Cas9, it targets that very part of the DNA that I know will ultimately be transcribed and translated into a protein that gives you blue eyes. And I'm going to chop it, and I'm going to rely on non-homologous end joining to bugger it up. Doesn't fix it very well, turns that gene off. I can now have a look and see how it's expressed, have a look at the phenotype. And that's what we can use CRISPR-Cas9 complex with non-homologous end joining for, to turn genes off. The other way, any questions?
No, keep going.
The other way that we can repair these double-stranded breaks is through what's called homology-directed repair. So HRD. No. HDR. Homology-directed repair. Now, this is where CRISPR becomes super interesting. So again, CRISPR-Cas9 comes in, chops both strands of DNA, but this time, remember we said earlier on that you actually have 2 copies of your DNA. So I may have just cut 2 strands of the one DNA, but you've got another one left. So what can happen is one of those busted strands goes into the other strand and uses it as a template to just fill in the gaps. So it's a more specific, accurate way of going, oh, this got cut. What nucleotides are we missing? Here's a template. Copy.
Would that be considered an allele? No.
An allele is, just a discrete copy of a gene. So we tend to have 2 alleles of every gene.
So one from mum, one from dad?
One from mum, one from dad.
So is that what it's doing?
Yes, so it's going to, let's just say we chopped mum's version that she gave you.
Yeah.
One of the broken strands jumps into dad's. It's all there, reads what nucleotides are missing, fixes the gap, then jumps back in to its broken DNA strand, and now it's used as a template to fix its original broken strand, and now it's fixed. And that's a very more specific, directed way of fixing the DNA to remove errors, right? So that it's still ultimately going to be a functional gene. But researchers are like, wait a minute. If it uses a template, why don't I just introduce whatever template I want? So I can create my own CRISPR RNA to guide it wherever it wants to go in the genome to make a very specific cut. And then I can add my own template to say, hey, fix it with this instead. Okay. And now I've just added a whole new gene into your DNA to what we call a performer knock-in.
That's interesting.
So I can now add a gene to be expressed. So I might go, you know what, Matt? You don't contain this particular gene. I want to see how it looks on you, the way it's expressed. I do this homology-directed repair using a template of a very specific gene. It adds it into your DNA, and I can watch it being expressed.
So basically what you can do is through this CRISPR technology that we've hijacked from the bacteria, the bacteria basically, through its own intrinsic system, is when it gets infected by a virus, the virus puts its genetic information in, which essentially will be incorporated into the bacteria's genome. But when that does occur, the bacteria can make RNA from that, which incorporates itself into a protein which we call Cas9. And when the bacteria gets infected again by that same virus, that complex comes along to the viral RNA or DNA? DNA. And what's in the tracer that's incorporated in the Cas9 will read a section... Spacer. Will read a section of the viral DNA and go, hang on, I've seen this before. What I'm going to do is I'm going to deactivate it and just chop it in half.
That's right.
Now, we're hijacking that system. So we basically take bacteria's Cas9
complex, the CRISPR-Cas9 complex.
But now we can maybe alter what's inside it. So that's the arms of the glasses.
In actual fact, there's 3 things we can alter, just to interrupt very quickly. We can alter the RNA template to tell it where we want it to go in the DNA. We can alter by adding a template to say, fix it with this, right? Or add this. And you can even alter the Cas9. And you can alter the Cas9 to break it in different ways.
Okay. And I think this is what they did, right? Because we're essentially removing machinery from a bacteria, it's, to us as humans, it's foreign proteins.
Yeah.
And so because we are taking fragments from bacteria that may come from families that are pathogenic to us, like a lot of these bacteria that they've done the research on, like Streptococcus or Staphylococcus, are pathogenic bacteria to us. So it's likely that humans have incorporated in our own genome, particularly our immune system, adaptive technologies that if we encounter a bacteria protein, we have antibodies against it.
Yeah.
So there's a possibility if we were to incorporate this CRISPR technology with the Cas9, the bean protein, that we already have antibodies to it. Yeah. And so that could be a limitation.
Yes, because we might say, hey, that shouldn't be there. That's bacterial. I'm going to attack it and destroy it.
And so that then negates the whole technology, right? Because
one of the limitations.
And the other one that I came across was instead of doing, double-stranded breaks, we can do more intricate breaks.
Yes, that's right. And that's by altering that Cas9. Or there's even other Cas proteins that can be used for these types of breaks. But yes, you can alter it to have many different types of breaks. The other beautiful thing about this CRISPR-Cas9 complex is that historically, traditionally, in molecular biology, for example, like in my PhD and my research, it was really difficult to add a single mutation, right, into a cell line, let alone multiple mutations discretely. Because the way that we couldn't be so direct and accurate each time. So it would take many attempts, and many times most of the cells didn't have the change that we wanted, and we had to pick and choose the cells that ended up having the mutation and propagate those. And you can only really do one at a time. Doing single nucleotide changes was really hard. Doing chunks was easier because you could just chop out and glue back in. But again, you couldn't do multiple areas at the same time.
Makes sense.
You can with CRISPR-Cas9. And the benefit here is how it's actually mostly being used at the moment is for screening. So when we think about cancers, we spoke earlier that cancer is a genetic disease and that often the mutations will occur either in tumor suppressors to inactivate them or oncogenes to amplify them or exaggerate them.
Or both.
Or both.
Or amongst others.
Amongst others, that's right. So what we can do, and this is an example that they've done recently, is they've taken colon cells and they've gone, okay, colon cancer, very common type of cancer. It's killing millions of people every single year. We don't fully understand what genes are involved, and not just that, not just the genes, but also what type of mutations in those genes result in this type of cancer. So CRISPR-Cas9, you can actually take those cells in a dish, for example, and you can incorporate many mutations in many genes in many different areas and have a look and look at the phenotype. How does it grow and develop? And does it look like the type of tumors or cancers that we see in vivo in a person with bowel cancer, for example? And so if we can recapitulate that or copy that in the lab, we know exactly what genes are involved, what types of mutations, where they are, and now we can target them to fix them. So that's what we need to do, is the screening. We can also then, once we create cells that are virtually identical to the tumor or cancer cells that we make, we can then throw certain chemicals or drugs onto them to, again, screen for successful treatments. And so this is how CRISPR-Cas9 is actually being used most commonly, is for screening.
So just within an in vitro environment.
That's right. Yes.
But,
so
but also having the capabilities. So instead of thinking it's solely being used in human models, you could also utilize it in animal models where you may have an idea of what the disease phenotype is. So this could be an Alzheimer's disease. This could be a Parkinson's disease. This could be Huntington's disease. This could be cancer. And you could manipulate the genes of a mouse model. Again, either a knock-in or something similar. And then you have...
Or knockout.
The closest that you can get is this, this disease model, like a bowel cancer model in a mouse, that then you could not only try to experiment diagnostically to how better can we screen for pre-cancer states, but also we could even do what kind of environments. So this could be just, cancer in many cases, like you said, is a genetically based disorder, but it could be from a background of an inherited gene mutation with an association of an environmental or a multitude of environmental stimuli, right?
Yes.
And so you could
people should know, sorry, super quick interruption, but people should know that regardless of the origin of the cancer or the earliest preceding cause, it's all genetic. So whether it may be inherited through a parent or whether it's an environmental thing like sun, chemical exposure, whatever, all the effects, all the things that result in cancer are genetically based.
Yeah, that's right.
Sorry.
So you then might have an individual that's, always going to get bowel cancer. Nothing that they could do about it. They're going to get it
Because it's just sitting there in the gene,
right, and it's
waiting for that time for it to express.
That's right. Eventually, it's going to come out. But if you expose it to a certain environment, and this is what could be studied within the mouse models, is you could maybe feed it a certain diet, or change the biota or the bacteria, of the gastrointestinal tract, and you might see this becomes protective, or this maybe brings the cancer out years ahead.
Absolutely.
And so then we could have a better idea of how the disease manifests, but also the treatment regime.
Yes. And I think finally, unless there's other things you want to say, which I'm sure there are, but one of the things, and this for me is what I believe to be one of the most promising aspects of the CRISPR-Cas9 system and its various iterations, which is going to be a multitude of, already are, is that I personally believe, and I might be wrong, don't hold me to this, but I personally believe that cancer will, within a generation, be a thing of the past. I personally think that and hope that my generation will be the last generation to die of cancer, because this technology is advancing so quickly, and its ability, children are being cured of certain blood cancers because of CRISPR technology. That was an impossibility a decade ago. And so it's only been a handful of years since CRISPR. Honestly, it was I think it was 2012, researchers started to say we might be able to use CRISPR as a gene editing tool for cancers. So that is not much more than a decade ago. And now we've cured people of cancers. And so I personally think that my generation, our generation, the listeners' generation, unfortunately, and fortunately at the same time, will be the last generation to die of cancer, because we will be able to screen people, let's just say at birth, and go, oh, look, you've got these particular genes with these particular mutations in them. It's not going to be necessarily definitive. Some might be, like certain BRCA mutations. You've got such a high likelihood of developing breast cancer from those, for example, and other types of cancers. And might be able to go, look, there is a mutation that we're so confident that at some point you're probably going to develop a cancer from that. Let's use CRISPR and just change it. And then you will not develop that type of cancer. Now, it might be a little bit different when it comes to things like skin cancers, where you get exposed to UV light in your 30s, and then a few years later you start to develop a skin cancer of some sort. But there's no reason why CRISPR technologies won't be able to address that once the cancer is then present.
Yeah, and that kind of goes to, so we've understood today how CRISPR in principle works. But
yes.
But sometimes the application is what limits the type of therapeutics it can be used for.
Yeah.
So by and large, what I've seen is most of the clinical trials that have been using CRISPR has been probably more successful in what we call ex vivo approaches. Or
take the tissue, put it in a dish.
So it's the way it's delivered. So I'll give you an example. One of the first ex vivo clinical trials that have been used, and it's getting pretty close to releasing the stage 3 trial results. They still have to follow these patients for longer periods, but quite promising results where they have individuals, let's say, with a type of anemia. So this could be a thalassemia or a sickle cell anemia. And so
genetically caused diseases.
This is where there's a alteration in the hemoglobin molecule of the red blood cell, which changes the way that oxygen can bind to, making it less efficient, but also the shape of the red blood cell, which can then lead to certain microthrombi, which is not only painful but potentially is deadly for the individual. And so what they can do is they can remove blood from the individual, and in the blood, peripheral blood smear, they can isolate certain let's say progenitor stem cells. So these are cells that are likely to have children that will become red blood cells. So they're not mature yet.
And they contain the mutations for these diseases?
Yep.
All right.
And so then...
But they also have the potential to create progeny.
That's right. So that when you're a mature red blood cell, you can't do anything further with it. Okay, it's stuck. It's going to be how it is. Okay, it's going to survive for 120 days, and then it's going to die. But if you can get a stem cell, it can have multiple generations. So if you can alter that, then you may have benefit in what lineages it produces.
You alter that, you alter the generations.
That's right. So they can pull these blood cells out. They can do a CRISPR approach to it, so they can change the hemoglobin that's dysfunctional, and they can express a different type of hemoglobin, which they call a fetal hemoglobin, which is the hemoglobin that we use when we're an embryo.
Right.
Okay. But that kind of switches phenotype when we're adults.
Yep.
But unfortunately, the diseases, that's the diseased one for these guys.
Okay.
So they alter it, then they put it back into the individual, but at the same time, they kind of ablate the rest of the stem cells in their bone marrow.
Right. So they use radiotherapy to just...
Or chemotherapy.
to just destroy all the cells within the bone marrow.
Correct.
Because they're going, look, they're bad.
Yes.
Let's knock them out.
Yep.
We're going to introduce
or
the
just kill
edited.
them. Rather than say knockout, because we've already used that term.
True.
Let's just say kill them off.
Okay. Yep.
And these progenitor stem cells then can go and reside back into the bone marrow, take hold again,
become resident stem cells.
Yep. And so it's shown some promise that not only do these stem cells recolonize the bone marrow, but then the percentage of hemoglobin in the circulation now is 60% fetal hemoglobin.
Right. And that's not a problem?
Not a problem. It's not as potentially efficient as the adult hemoglobin, but it's better than what that...
Sickle cell anemia.
Correct.
Yeah. Amazing.
So that's an example. So that's an
So we don't have stage 3 results yet?
Well, the clinical trial was published in New England Journal of Medicine.
When? Do you know?
2021.
Okay. And?
Oh, very promising.
Awesome.
But it still have to follow out the stage 3 to just ensure that there's no off-target effects.
Right. So what do you think is the future of this? Do you agree with my...
The other thing I'll just say before I get to
political.
is the advantages of doing an ex vivo approach is that you can be much more tight and regulated in your manipulation, because the way you manipulate is outside the body.
Yeah.
So you can be much more sure that what you've manipulated with the cells before you put them back in is safe and accurate.
Yeah.
But the limitation by the ex vivo approach is, well, it may be difficult to get those cells that you're going to put back in to the target that you want it to reach.
And you're not editing everything.
Right.
You're editing a subpopulation. And unless you ablate or destroy, like you said, all the other cells that are still present endogenously, it's sort of like a competition.
Yes.
And so, but I think we'll find ways around this.
But this then goes to the in vivo approach.
Okay.
Where you can actually put your CRISPR in either through viruses or even through similar to what we saw with mRNA COVID vaccines.
mRNA, yeah.
Is that what I said?
You said mRNA.
Okay. I just said the N very quickly.
Okay.
That you tell your cells to make the CRISPR technology for you.
Yeah.
And it does the manipulation in that group of tissue.
Unfortunately, no one trusts mRNA vaccines anymore.
Not at the moment.
Which is sad.
So the benefit of this approach...
I'll take it.
...is it's in a way less invasive because you don't have to pull cells out that can be from discrete locations.
Yeah.
You're just putting it in. A greater, wider range of target approaches, but the challenge is how do we restrict it to just doing that one thing, and it may produce a lot more off-target effects.
Yeah. So what do you predict for the future with this type of technology? How do you think it's going to... Do you think it will be... How significantly beneficial or not do you think it will be?
Well, I think the one challenge with cancer that we have to remember is cancer in itself is its own organism. Yeah. And so it's not a homogenized group of cells. All those cells are trying to survive themselves, right? And so this is part of the problem with
They're really not your cells anymore, are they?
That's right. They're like a separate organism.
Yeah.
And so they're trying to survive within you. And this is part of the problem with, cancer therapies, is they are evolving to that therapy. So as we're throwing chemotherapy at it, whether it's the traditional ones or even immunotherapy and so forth, that they're trying to adapt for their own survival. And so their populations are changing. So even if we are trying to figure out, like you're talking about with colon cancer, ways of screening and identifying, then maybe they're altering as well. And so we have to keep in pace with them.
Maybe. But my rebuttal to that is we will be with CRISPR editing the earliest known basic cause of the cancer, which is the DNA. And with that edited,
especially if it's preventative.
Yeah, I think. Yeah. I mean, that's going to be its biggest application. Will be as a preventative technology. You screen, you identify, you alter, you mitigate the requirement.
Or even an alternative to that is, in a way, we could look at cancer therapeutics a bit like vaccine technology, where we know that we use vaccines, inoculate for your immune system to then target, that virus when it comes into your body or that bacteria. We can use the same approach where, and this is, I think, the CAR-T,
yeah.
Where
chimeric antigen receptor technology.
Yeah, so
or T cells, sorry.
And there's like 250 odd clinical trials in this space.
Yeah.
Where basically what they're doing is getting T cells, maybe let's say the cytotoxic T cell, which is the killer of the T cells.
This is that example of the young girl cured by leukemia that I was telling you about. They use the CAR-T technology.
Right. So what they can do is, because the T cells are adaptive immune systems, so they are specific to usually the pathogen that you're being infected with, you could do the same approach but do it to the cancer cell. Find
to the T cell.
The antigen on the cancer cell.
Yeah. So you said do it to the cancer cell, but you mean use the CRISPR technology for the T cell.
Correct.
Yeah.
So you need to find, like we spoke about, we need to find the particulars of that cancer.
Yeah.
Okay. So that could be done in an animal model, or that could be done by taking the cells of the cancer out and going, well, what's the identifying factor of this particular cancer that's different to your own cells? Because you don't want to kill your own cells off.
Yeah, so our body has little flags on its surface to say, "Hey, I belong to you." Even though your cancerous cells are at least originally were your cells, they do express certain protein flags that make them distinguishable from other cells. Right. So sort of like cancer flags. And so what you're saying is that you could train the T cells to recognize those flags.
Maybe like, say, where's Wally?
Yeah.
Right. So where's Wally wears this red and white beanie, right?
Yeah.
And so that's makes him distinguished to the rest of that really busy,
picture book scene
there. Yeah. And so you can tell your T cell, this is what you need to look for. And it becomes an assassin. You release it back into the body, and it could be done in the blood through IV. And then it goes. And then
attacks and destroys.
And once it comes across that particular antigen, then what a cytotoxic T cell does is just kills the cell.
Yeah.
And then that could also be an approach.
So and it is, right? Like we've utilized it. So I think the great thing about this CRISPR technology and its various iterations are it's wide reaching, right? It's broad. It's going to be utilized in a number of different ways. It's not going to be a one-way approach. It's going to be utilized in many different ways. And again, I'm really excited. I think this is going to be a huge shift in disease burden for the future. I mean, cancer is still one of the biggest killers.
Number 3 in Australia.
Yeah.
Or number 2.
Outside of cardiovascular and metabolic disease. Cancer.
Yeah.
Well, and COVID over the past 2 years, that's been a big killer. But yeah, it's exciting. And I hope everyone found this interesting.
I don't want to go too deep down this path, but it does raise ethical concerns.
Yeah.
Particularly if you are starting to look at manipulating genes in germinal cell lines.
Yes.
So we've spoken all today about somatic cell.
Yep.
But there has been at least one example I've heard in 2012 China, was that right? 2018.
Yep, 18.
Where a scientist altered, sperm cells for HIV resistant genes, and then through IVF fertilized the oocytes and then introduced those blastocysts into a surrogate, and then
She gave birth to twin girls.
and that's as much as we know.
That's it.
It hasn't been peer reviewed, and I don't think the individuals have been followed. But this does, and I think this is the biggest pushback that the other scientists throughout the world gave, was the ethical concerns around this, because it does then not only impact that lineage, but it could alter lineages to come.
Absolutely. And
Because the off-target effects not only is the hardware change, but also you could have even epigenetic changes, right?
that's right. So, yeah, this researcher by the name of He Jiankui, like you said, gene edited cells so that they were more HIV resistant, created embryos or blastocysts, implanted them into a woman. That woman gave birth to twins, and now these twin girls have they're walking around in society. This is 2018, so what would they be? 4 years old now, 5 years old. They're walking around. We don't know who they are. They've been de-identified. I'm sure the Chinese government knows who they are and where they are and keeping eyes on them. But he, being his name, he went to prison for 3 years, only got out, last year or 2021 or something, and now works for a biotech firm doing similar stuff. But there's a huge ethical concern here, because if he was able to do it, what does that mean for... look, he may have had the best of intentions, right, looking at HIV resistance, but what if, for example, you want to change a trait that isn't necessarily there to reduce somebody's risk of disease?
And that's the movie Attica, right?
Yes.
Which is basically germal cell manipulation.
Yes. To produce superhumans.
Superhumans.
Yeah.
So you can change traits that you kind of wish.
It's dated, but it's good. And I didn't realize for many years that Attica is just...
the genetic code.
Yeah, ATTAC. Yeah, anyway. So, yeah, look, he went to prison for 3 years, got fined like $500,000, and now works for a biotech company doing genetic engineering. And the Chinese government have... This is the thing, you need to have really strict ethical and moral rules and regulations around the way this works. And most countries really do, but I'm unsure as to how this happened, because there's many steps to this process. And people who aren't in the research field may think, he's just a single scientist doing it in his lab. No one knew. He did it all by himself, like in the movies. That's impossible.
Yeah.
It's impossible to do this without doing it in a lab that other people are accessing, that other people are in, and without using the expertise of other individuals. As a researcher, he may have been able to do the genetic manipulations with CRISPR, but he definitely wasn't performing the IVF, right?
Yeah.
He wouldn't have been trained to do that. That's a very specific field. And as we all know, that is not just as simple as injecting some cells and saying, off you go. There's hormone therapy treatments. There's, this is a process that would have involved...
Well, you have to extract the eggs. You have to do the fertilization outside, and then you have to do the implantation. This
would have involved at least a dozen people. At least. In a big, well-funded lab. Multiple labs, actually, because they all wouldn't have done the IVF. And so, I mean, if you sit back and you think this is not something that just happens because of one lone weird rogue scientist, it's not the case. I mean, it may have been how they sold it and said, oh, yeah, look, he's been reprimanded. He's in prison. Don't worry about it. He went rogue. This is bigger than one individual.
Yeah.
Anyway.
And just to reassure the listeners...
we're not doing this.
This is not regularly done in the Todorovich lab. No. When we do research either with animals or humans, it's very tightly regulated with ethics, animal ethics, human ethics. And
Matt, how long does it take for you to do an animal ethics application?
Well, my research is in neuroscience, and I look at just nerve repair, and it's heavily scrutinized now, which is
good
for good reason.
Good,
but for the number of animal use, why you need to use animals, why you need to use that number, why is that number robust, etc., 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 and all the ethical interventions that will be used to ensure that the animal is not suffering and so forth. And that's not with animals that are even approaching genetic modification. And so when you start to do genetically modified organisms, it's even...
So a lot of my work was GMO.
And you have to...
I didn't do animal work. I just did cell work, right, with genetically modified cells. The approval process, storing the cells... Transporting applications.
And even disposal. Having people come in every X amount of months and reviewing your lab to say, hey, where's everything being stored and where's this and where's that? I mean, it's a whole big thing, as it should be. Yeah.
But just so the listener knows, this is extremely tightly regulated. But in saying that, if done well and done rigorously, this will be life-saving.
Yes.
And that, my friend, is CRISPR.
Oh, quickly.
Yeah.
No mailbag this week.
What happened to it?
It must have had a hole at the bottom of the bag, and then the mail dropped out.
Really?
So no emails.
No. Well, in actual fact, we do have some, but we just didn't get them sorted.
No, I haven't received any. Did you get some?
Yes.
Okay.
But I didn't prepare them.
All right, we'll leave that for the next one.
Yes.
Is there any final remarks you need to make?
Yeah, I should. I should have probably said it at the very beginning of the podcast. If you want to support us, you don't need to give us any money. I mean, if you want to, that's actually fine. But no.
In a suitcase?
Yes.
At what corner of what street?
1 p.m. on the corner of 5th and 3rd on the 17th. Subscribe to both our podcast and our YouTube channel. Please watch, like, view, comment, give us 5 stars. Do all the nice, kind things that you would do to help support somebody that you like. If you want to contact us, send us an email. Our email address is gubiosciences@gmail.com. So gubiosciences@gmail.com. Or you could just go to our website, which is drmatt.drmike.com.au. So DRMIK.
I'm just checking that now just to make sure it is working.
DRMATTDRMIKE.com.au.
And on the homepage, there's an area that says reach out.
There you go. So reach out, send us an email, ask us a question, give us feedback. We do this because we love doing it and we like helping people understand the human body and how everything works. Apart from that, I mean, you can follow us on social media at Dr. Mike Todorovich on all platforms. Don't worry about following Matt. He doesn't have social media, and even if he did, he's boring.
That's true.
So it's pointless. I barely like doing these podcasts with him. So apart from that, Matt, thanks for nothing. And listener, we love you.
I'm off to eat some crisps.
Please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovich, at DRMIKETODOROVIC. Speak to you soon.